One of the major things we see in adipose tissue from cows with ketosis is that even with subclinical ketosis, is that metabolically that tissue is different, even though the cows are not yet demonstrating any clinical sign of ketosis. So I think it is in our transcriptome data sets, for example, we have been doing a lot of RNA sequencing, single cell sequencing. We see that there is like this enrichment for genes that are encoding extracellular matrix remodeling, more changes in immune signaling within adipose tissue of cows with ketosis. So what this suggests is that there is also a structural and inflammatory adaptation alongside increased fat mobilization.
Hello everyone, this is Luis Ferreiro with the Dairy Nutrition Blackbelt Podcast, and today we want to discuss about the importance of adipose tissue to transition cows. And to shed some light into this topic, we have Dr. Clarissa Strieder-Barboza, Assistant Professor in Veterinary Science at Texas Tech. Clarissa, thank you so much for joining us today. I know that you have been working a lot in this area and will truly help us to better navigate this key topic for dairy producers, right? So let's start exactly with that. Why dairy producers should care about adipose tissue in dairy cows?
Hi, Luis. Thank you very much for the invitation. I'm very happy to be here sharing some of what we have been learning in the past few years in this topic of research. So why is adipose tissue important? I think historically, everybody thought that adipose tissue was just a passive organ, just storing energy and fat. But nowadays, we know that adipose tissue is a major endocrine organ that is synthesizing cytokines, adipokines, is modulating metabolism, inflammation, and also disease risk. So that's why it is very important. We know, for example, transition cows and mostly in early lactation, these cows can mobilize up to 1 kilogram or more than 2 pounds of fat per day, right? So the way they do that is through the mobilization of non-esterified fatty acids, or the famous NIFA that we all talk about. And these NIFAs can influence not only liver function, but also immune response, milk production, reproduction as well. And also, if it's in excess, it can lead to disease like fatty liver, also increase insulin resistance, immune dysfunction, and of course, increasing the risk of ketosis, which we know is very prevalent. So what our research has shown is that adipose tissue does not respond passively to negative energy balance. What we have discovered is that adipose tissue remodels. There is structural changes in the extracellular matrix, which is that non-cellular part of the adipose tissue, as well as immune infiltration and the changes in metabolism. So all these modify how cows handle stress in the transition period.
Looking to maximize your herd's potential? Elevate performance with Kemin's cutting-edge encapsulation technologies, including rumen-protected choline, methionine, and lysine. Kemin's advanced choline and amino acid technologies ensure precise nutrient delivery, boosting milk yield and enhancing herd health. Trust Kemin, the experts in encapsulation, to take your herd to the next level. Learn more today at kemin.com/dairy.
So clearly, there's a lot going on, right? And the simple fact that adipose tissue is being mobilized can obviously create a cascade of chain reaction, right? So if we go back to this topic about causing metabolic disorders and that interaction between you mentioned a little bit about negative energy balance. Tell us a little bit more about what happens metabolically with the transition cow especially related to this potential influence on ketosis?
Yes. So ketosis is not just a liver disorder. It starts in adipose tissue regulation of lipolysis. Because during this early lactation, there is that drop in insulin. There is a higher sensitivity of the adipose tissue to catecholamines, which will trigger higher lipolysis. With higher lipolysis, there is more NIFA in circulation. And then there will be, or there will be not, more production of ketone bodies, which characterizes ketosis, right? So in cows with ketosis, there is more lipolysis. So coming from adipose tissue, dysfunction on regulating that lipolysis, right? Insulin signaling is impaired. So the cows not only have low insulin, but also the insulin signaling or sensitivity in adipose tissue and other peripheral organs such as muscle is also impaired. There is also more inflammation in the adipose tissue, also reflecting in systemic inflammation. And there is this extracellular matrix remodeling of the tissue. The tissue is reorganizing itself. So that also may cause metabolic disorders. So one of the major things we see in adipose tissue from cows with ketosis is that even with subclinical ketosis, is that metabolically that tissue is different, even though the cows are not yet demonstrating any clinical sign of ketosis. So I think it is in our transcriptome data sets, for example, we have been doing a lot of RNA sequencing, single cell sequencing. We see that there is like this enrichment for genes that are encoding extracellular matrix remodeling, more changes in immune signaling within adipose tissue of cows with ketosis. So what this suggests is that there is also a structural and inflammatory adaptation alongside increased fat mobilization.
Priority IAC, the only company bringing the fields of microbiology and nutrition together and the first ingredient in animal diets. Priority provides a more cost-effective and easier approach to nutrition. We recognize that not all bacteria are created equal. The specific strain truly matters. Priority IAC selects unique strains to provide the best performance in animals, and that's why we call these strains smart bacteria. Founded by Richard Brunig, helping to improve animal health one herd at a time.
So you mentioned a little bit about your transcriptome research and how does that copy with some of the metabolic disorders. And I know you have been using some very advanced genetic tools in your lab. So tell us a little bit more about what are these tools and what do they reveal about those issues in the transition cows?
Yes. So adipose tissue is actually a incredible universe of specialized cell types. Not only adipocytes, right? And again, historically, we always thought adipose tissue was just composed by adipocytes, which is not true. And we know this based on human, mouse, and also now we know based on cows studies as well. So if you compare the single cell of bulk RNA sequencing, which is most known compared to a single cell. In the bulk RNA sequencing, you average the signal across all millions of cells in the tissue, right? So it's just an average of gene expression. With single cell or single nucleus sequencing, which is what I do, because with single nucleus, I can also detect signal of these large cells, which are adipocytes, which are not detected in single cell. It lets you see the subtypes of cells. So we see, for example, adipocyte subtypes. We know that not all adipocytes are the same. We see the macrophage populations. we see the fibroadipogenic progenitors, which are progenitors that can go to the adipogenic pathway or can just remain fibroblasts. We see populations of vascular cells, including endothelial cells, smooth muscle cells. And of course, we see other immune cells and the patterns of these immune cells. So in summary, you can characterize and see the gene expression of each cell type and subtype within the adipose tissue. So instead of asking or knowing what adipose tissue is doing overall, in average, we can now know what each cell type is doing. So from our recent analysis, we have learned, for example, very interestingly, that there is a specific adipocyte subtype that is in higher frequency in animals with subclinical ketosis. And it's only present in visceral adipose tissue, not in subcutaneous adipose tissue. It is enriched for leptin and is also enriched in extracellular matrix genes, which may imply that some subtypes of cells are also reacting to ketosis within adipose tissue. So that tells us that adipocytes themselves specialize during metabolic stress as well.
Today's episode is sponsored by S Milko Inc., baby animal nutrition specialist focused on high-quality milk replacers. S Milko works closely with producers, nutritionists, and veterinarians to support healthy starts, strong performance, and consistent results in calves. With carefully formulated products and a commitment to quality, S Milko helps set the foundation for lifelong animal success. S Milko Inc., setting the nutritional foundation for strong, healthy animals from day one. Learn more at esmilco.com.
So, thinking about from a dairy nutrition and management perspective, right? How can we use this type of information in the future to change what we do with dairy cows and make sure we either prevent or solve those issues associated with metabolic disorders?
Yes. So I think the goal in the transition cow road is improving metabolic adaptation or metabolic resilience, not just preventing energy deficit, right? So understanding how the adipose tissue works or the physiology and biology of adipose tissue throughout the transition period, we could identify cows that are at higher risk for developing diseases earlier. And we are now developing a study that we start at the dry off until early lactation. We can, of course, use this data to refine transition diets. We can try to improve the inflammatory balance. We now know that chronic state of inflammation in transition cows is also associated with higher risk of diseases and lower performance. We can support insulin sensitivity, and therefore we can also reduce the prevalence of ketosis. So if we understand how adipose tissue adapts or fails to adapt during the transition period, we can design these nutrition strategies that may improve metabolic adaptation instead of only increasing the energy supply. And we have some examples of that, or strategies actually that have been used nowadays that also involve adipose tissue health. For example, we know some strategies that target insulin responsiveness and regulation of lipolysis. We know about the use of omega-3 fatty acids, antioxidants, and also methyl donors that are regulating or enhancing that anti-inflammatory nutritional support. And we also know of novel research that are revealing new biomarkers as, for example, adipokines that are produced by adipose tissue and could be used to detect disease. So one area that we're actually studying now that we are interested is how the catecholamine metabolism inside of the adipose tissue regulates fat mobilization. And my PhD student, Bridger Sparks, is working with a neuro-like cell type that we think that degrades or modulate this availability of catecholamines inside of adipose tissue. So, for example, in the future, I believe that we could target specific cell types within adipose tissue to modulate lipolysis and disease development.
Certainly very intriguing, and hopefully we'll keep finding solutions, right, to this issue that dairy producers certainly face and will continue to face, right? Clearly it's pure physiology. But thank you, Clarissa. I really appreciate you joining us today and sharing all this information with us. I'm sure that people at home will truly enjoy seeing all this information, trying to elucidate what comes next for them so they can be prepared for that whenever those solutions arrive to the industry. Thank you at home for joining us, and I hope to see you soon.
Thank you, Luis.